All right, with that, we'll go ahead and get going. So, Peter van der Plas has a background in histology and immunocytochemistry. He joined Orion in 1991, where he's worked alongside Dr. Jan Lunesen to develop and test advanced immunoreagents and to design the Orion masterclasses and in-house workshops on Immuno Gold Silver Staining. Peter's a seasoned educator and has shared his knowledge at numerous international microscopy conferences, and we are delighted to have him join us today to share his insights on Immuno Gold Silver Staining, everybody's most beloved technique. Peter, thank you so much for being here today. You have the floor.
Okay, thank you, James, for your kind introduction. And I will take over the floor then and just go towards I can't have my slides working, so you still...
You might have to click on your, depending on where you're clicking, you might have to, if you have like a PowerPoint with notes, you might have to click on the original PowerPoint slides.
Yeah, that's the
back
one.
on.
That's the one, okay. Okay, in today's webinar, I will present information on pre-embedding Immuno Gold Silver Staining. What is the difference with on-section Immuno Gold Staining, a more conventional method to detect and visualize antigens on the EM level? What are the advantages of pre-embedding labeling? What are the requirements for good to decent results, both with respect to labeling density, but also morphology? And what are the limitations of the method, because they are also there. So in Immuno Gold staining, Immuno Gold Silver Staining, of course, the Immuno Gold reagent is the key reagent that you're using. And colloidal gold is a marker particle. And that's an interesting characteristic, because when you have a particle and you can visualize that particle in an electron microscope, or in the microscope as such, then this means that you have the basis of a very sensitive system. When you see the gold particle, you know the antibody is there, and when the antigen has reacted, has detected the antigen, then you have a very sensitive system. In principle, you have the option to visualize a single antigens. That makes it very sensitive, of course. The particle is electron dense, it emits secondary electrons, so you can both use it in the transmission electron microscope and the scanning electron microscope. Another interesting characteristic is that it reflects light, it depolarizes light, and that makes it possible to use gold nanoparticles as marker molecules also in the light microscope. So you can visualize single particles in the light microscope, having an intermediate in between transmission electron microscopy and bright field light microscopy. So you can use a polarizing microscope for this. You can use the confocal for this. Well known, of course, for people or researchers that are working with gold conjugates, is that gold particles can be prepared in a wide range of diameters. So you can do multiple labeling studies using gold nanoparticle conjugates. Again, what I said, the particulate nature results in improved resolution. When your particle is linked directly to the primary itself, then the particle is very close to the antigen, and that improves resolution. Another interesting characteristic important for Immuno Gold Silver Staining is that these particles can act as catalytic nucleoli for the deposition of silver. So if you have silver ions close to your gold nanoparticles, you have an electron donor, then the particle acts as a catalyzer, and you get a specific deposition of metallic silver on your gold nanoparticles. And that can be used to visualize antigens on different levels, as I already mentioned. So in this slide, you see in the left image over here a Tokiyasu cryo section. And in this section, COP2, a coat protein present on the vesicle membrane, is being demonstrated with a polyclonal antibody against COP2 and visualized with a ultra-small gold nanoparticle conjugated, Fab fragmented against a rabbit IgG, silver enhanced for visualization. So particles are lying very close together to the area where the antigen is located. When you go on with silver enhancement, particles may fuse, and then you will end up with a silver deposit, and that deposit is visible in the light microscope. So you can use Immuno Gold Silver Staining also in immunolight microscopy. You have a dense brown to black signal which can easily be combined with hemotoxylin and using counterstaining methods, or for instance also with nuclear fast red. Very high contrast between the signal, specific signal, and the overall morphology of your specimen. In this image, E-cadherin, cell adhesion protein, is being demonstrated using a monoclonal antibody against E-cadherin and goat anti-mouse IgG, again linked to ultra-small gold nanoparticles, silver enhanced to obtain your end signal. You can also use it in diagnostic tests, immunoblots. In the right panel, on the right image, you see the demonstration of tubulin on a blotting membrane. Both alpha and beta tubulin are being demonstrated, are visualized with this two-step method using a polyclonal antibody against tubulin and a detection with goat anti-rabbit ultra-small and silver enhancement. So due to the fact that gold nanoparticles can act as catalytic nuclei, you can use this method, Immuno Gold Silver Staining, on all levels of immunostaining, being EM, LM, and diagnostic tests. Look, what is happening? Do you still see me? Yes. In general, a two-step immunodetection method is being used because this has the wider application. You can use or demonstrate your antigens using a specific polyclonal or monoclonal antibody or an antibody fragment and visualize, detect this specific primary with either antibodies conjugated to gold, antibody fragments conjugated to gold, or an immunoreactive protein like protein G or protein A or stratavidin to visualize your primary antibody. This gives in general adequate resolution, so your particle is still close to your antigen. And depending on the system that you're using, you can have 1 to 4 gold nanoparticles per antigen. So these polyclonal antibodies that are linked to the surface of a gold nanoparticle will detect different antigens on the primary. That means that depending, and that is in relation to the gold nanoparticle size that you are using, you can have more than one particle per primary. This will give an increase in the sensitivity of detection, so you see more particles per primary, but you still recognize or detect only one antigen. So that's the difference with, for instance, protein A gold, which binds to the SC part of the primary antibody, and there you have this 1-to-1 ratio between antigen and particle number. What I find important in a two-step method is that it's easy to distinguish specific reaction with non-specific reaction. So you can easily see the difference between the reaction with your antibody and background levels by including a negative control where you leave out the primary antibody and only incubate with your specific gold reagent. If this negative control is clean, then you know your system works well. So in general, these two-step methods are being used also with other immuno systems or immunodetection systems. You will see that the two-step detection system is most often used because it's easy. It's easy to use. If you really want to have your particle as close as possible to the antigen, so if you want to obtain the highest resolution, then go towards a conjugation of your primary directly to the gold nanoparticles, or even use a fab fragment of your primary and link that, conjugate that directly to your antibody. So I already mentioned that in the beginning, that depending on the application area, you optimize your silver enhancement system and your silver enhancement time. So for EM, it's important that you have an efficient system, that your gold-silver particles are homogeneous. And that's something that you can obtain with the reagent that we developed for electron microscopy. If you go on with silver enhancement, then in time particles will grow and in time particles will fuse. And then you end up with a silver deposit. And that's suitable for research projects that you use, where you use LM evaluation, or when you're using your eye, so your bloods, for instance, on bloods. And there it's important that your deposit is intensely colored, is dark, has high contrast. And that's something that you can obtain with the Argent SE intense systems. So we have developed two systems, one especially for homogeneity and efficiency, which is called Argent SEEM, and one that gives contrast, which is suitable for the use in light microscopy and on bloods, and that is called Argent SE intense. So your particle, your gold particle, is the catalyzer of this system. So you have a mixture of silver ions and an electron donor, and due to the fact that your gold particles carries a negative surface charge, a highly concentration of negative charge, then this negative charge allows an efficient electron transfer from the reducing agent towards the silver ions that have bound to the surface of your metal particle. And then in time, the diameter of your particles will grow, so you get deposition of silver on the metal surface. In time it will grow, your particles will grow, they will fuse in time, and then you get this densely colored, densely contrasted of the metallic silver. So this is a method that you all know, the post-embedding method, where you have specimens, for instance, cells in culture, small tissue blocks. They will be fixed, chemically fixed, to obtain a reasonable to good morphology. You embed your specimens in a plastic, you section, and then you start with your immunodetection method. A method, the conventional approach that is often being used in immunocytochemistry. The advantage of the method is that you get adequate preservation of ultrastructure. Another advantage, and I feel that's the most important advantage of this method, is that you can obtain many sections from one single specimen. So in principle, you have the option to detect a lot of different antigens from one single specimen. The disadvantage of the method is the following. If you have a plastic section, because most often plastics are being used for immunodetection, then only those antigens that are on the surface, so really available on the surface of your section, will be visualized, will be available to be detected with primary antibodies and visualized with gold nanoparticles and silver enhancement. That means that you only have a small portion of your antigens available for visualization, for detection. In this example, where I labeled actin in low-equal embedded vascular smooth muscle cells, you see the actin fiber, the stress fiber, which is all actin, labeled with the antibody and visualized with the gold nanoparticles. And then you see that only a small portion of the actin is really labeled with particles. So with post-embedding immunocytochemistry, immuno gold silver staining, you can only label those antigens that are present at the surface of the specimen. And that's a disadvantage. Special plastics are needed. Special equipment is needed. The specimen preparation time is relatively long. So you may have the option to improve labeling density by leaving out the plastic. So work on a hydrated specimen. And the alternative is Tokuyasu sectioning. But also in this hydrated system, you can only label the surface of your section when using conventional gold nanoparticles. So this is an example where I incubated Chinese hamster's ovary cells, a semi-thick section, so of a hydrated section, Tokuyasu section, with a tubulin, an antitubulin primary antibody, and I visualized this primary antibody, so the tubulin, with a goat anti-rabbit 5 nanometer gold nanometer conjugate. I re-embedded the section in plastic and I prepared cross sections. So then you see a cross section through this Tokuyasu section, where you only see label on the surface of the section. This is the chromosome, this is the mitotic spindle figure, so this is all tubulin, but you only see label on top. So you miss a lot of antigen, a lot of sensitivity when working on sections. Not only plastic sections, but also on cryo sections, a hydrated section, when you're using large gold nanoparticle conjugates. The gold diameter may have or may play a critical role in this. But you do see if you when you go down with the gold particle diameter, then labeling density will increase when the diameter of your gold particle decreases. That's something that is well known and is being published in literature too. So smaller particles give higher labeling densities due to the fact that there is less steric hindrance, and also due to the fact that there is less repulsion. Gold particles, I already mentioned that earlier, carry a negative charge, so they will have difficulties to get in close proximity to each other. And the smaller the gold nanoparticle is, the easier it is to get them into close contact. So the smaller the particles are, the higher the labeling density will be. But what about penetration into hydrated specimen? Do you have advantages when using a small particle conjugate on a hydrated specimen, like for instance this Tokuyasu cryo section? Because when you're using small gold nanoparticle conjugates, then you have something more or less similar to what you have when you are incubating with a fluorescent conjugate or an enzyme conjugate. You have an antibody conjugated to one or more gold nanoparticles. And that is different from the conventional gold nanoparticle conjugates that you can directly see in the electron microscope. So the ones the conjugates that are based or prepared from with 10 nanometer or 15 nanometer gold nanoparticles. There you have a gold nanoparticle coated with a certain number of antibody molecules. So that makes the conjugate not only larger, but also well, less flexible. The antibody is less flexible because it's really linked towards the gold nanoparticle surface. And this ultra small conjugate concept is something that is really similar to what you have with immunofluorescent or immunoenzymatic conjugates. So this may well possibly be beneficial with respect to penetration into specimens, into cells, into cryosections, into hydrated specimens in general. And yes, these small conjugates have the benefit that they are very well able to penetrate into hydrated specimens. Here, this example of these Tokuyasu cryosections, now labeled with the polyclonal antibody and detected with goat anti-rabbit ultra small and silver enhancement. So not only labeling on top of the specimen, of the section, but also in the central part of the section. So labeling on top, but also the tubulin within the section is being labeled. So what about the pre-embedding method? Where you have your specimen, you start with fixation, with chemical fixation. After fixation, you detect your antigen with a primary antibody and with the ultra-small gold reagent. And after immunolabeling, you further process for either LM or EM observation. A setup that is similar to an immunofluorescent method or an immunoenzymatic method. Because you already have performed immunolabeling before processing for EM, so before embedding in plastic, you do not need specific plastics that are compatible with antigenicity. You can just switch towards an epoxy resin. So this setup is very simple and straightforward. You don't need any special plastics or sectioning techniques. The other advantage is that you can already check if your reaction has worked before going to the EM. So before processing the specimens for electron microscopy. That makes the system flexible and easy. Don't worry initially about the morphology. Try to get a decent result with respect to the detection of your antigen. Optimize your morphology by adapting fixation. Check again in the light microscope if your reaction has worked, and if yes, then process those specimens for electron microscopy, microscopic evaluation. So no special plastics or sectioning techniques are needed. You are working with a hydrated specimen, something that is similar to immunofluorescent techniques. Is that advantageous for labeling density? Yes. Here you see the post-embedding immunolabeling result when detecting your actin with Immuno Gold and Silver on a plastic section. You can only label the surface. And remember, this is all antigen, this is all actin. In light microscopy, immunolight microscopy, and immunofluorescent approach, then you can label or visualize the complete stress fiber. So the reason again, you have a hydrated specimen, a cell that's being fixed, being permeabilized with Triton in this case, and then the actin is being labeled with an anti-actin antibody and a goat anti-rabbit secondary antibody linked to FITC in this case. If you do something similar with ultra small gold in silver enhancement, then you can see that the number of particles on this stress fiber has increased enormously. So your labeling density again increases due to the fact that you have a hydrated specimen. You immunolabel before you embed in plastic, before you section. So that is advantageous with respect to labeling density. In this panel, these two sections, I fixed in aldehyde, formaldehyde. I washed my specimens, my cells, my cultured cells. I permeabilized the cells with Triton X100, and then I performed the immunoreaction. And you can see that in this case, when using only paraformaldehyde and in combination with Triton, the signal is there, but morphology is suboptimal. So you wash out, due to the fact that you're using this detergent, you wash out a lot of your cytoplasmic constituents. So for morphology, this is not a good idea, but for this setup with paraformaldehyde and Triton, but with respect to labeling density, it is a good idea. So what can you do to improve your morphology and combine this improved morphology with still a good reactivity of your antibodies and a good labeling density with your ultra small gold and your silver enhancement. Specimen pretreatment is important in IGSS to combine good labeling density with a decent ultrastructure. And there are several options to do that, and you can already test this in light microscopy. The test that I performed in the past to see if there was options to improve morphology and still have a decent labeling density, so was on cover slip culture, so pre-embedding Immuno Gold Silver Staining, staining the detection of tubulin in cover slip cultures of glutaraldehyde fixed red kangaroo cells, so epithelial cells in culture. After fixation in glutaraldehyde, because I had a primary that with standard glutaraldehyde fixation, so still could recognize the tubulin after glutaraldehyde fixation. I treated my cells with sodium borohydride. Sodium borohydride is a chemical, a reagent that transforms or converts the Sheaths base that is being formed by the aldehyde fixation, so the binding of the aldehyde towards the proteinable amino acid present in the proteins, that converts this Sheaths base towards a single bond and thus stabilizes fixation. So after this sodium borohydride treatment, I used Triton X100 to permeabilize the cells. After treatment with Triton, I incubated with the primary, washed and incubated with the Coat Anti-Rabbit Ultra Small Gold Conjugate. Incubation time with the primary 1 hour, with the gold conjugate 2 hours. So this is an option that you also have with larger gold nanoparticles if you use your Triton during fixation and after chemical fixation. So you sort of wash out everything except of the cytoskeletal elements, and your morphology, and that's what you see on the right panel, is when using this treatment is horrible. So it's not the way to go for optimal results, but it is a way to visualize antigens at the LM level. But what can you do to improve your results? So what other treatments are optional that improve your morphology, your ultrastructure, without having a real negative effect on labeling density? So I compared different protocols and checked and compared labeling density with morphology. In this setup, the cover slip culture of red kangaroo cells was fixed in glyceraldehyde again, treated with sodium borohydride, and after sodium borohydride, I washed my cells and permeabilized them in Triton X100. So the fact that I left out Triton X100 during fixation already had a positive effect on the morphology, on the ultrastructure, without having a negative effect on labeling density. So you see your cells, you see the tubulin fibers nicely colored, so intensely colored by the silver enhancement procedure. And when you switch or when you embed these cells in plastic and you section, then you can see your label. The particles are also on the tubulin fibers over here. But morphology is still suboptimal using this setup. So I can also compare different procedures. So for instance, also saponin, which you can also see in literature often being used in pre-embedding. Again, structure better and still a nice labeling density. So the tubulin fibers were nicely colored with gold and gold silver and silver enhanced. This last panel, I left out the permeabilization and only treated my specimens after aldehyde fixation with sodium borohydride. So no detergents being used after fixation, after sodium borohydride treatment. Directly incubated with primary antibody, ultra small gold in the second step, and then silver enhanced to visualize the signal in the light microscope. This is what I obtained. So I could see areas where the tubulin fibers were being stained, were being labeled, but there were other areas where I could see the cell, but no label was present in those areas. Morphology was nice, by the way. So initially I was a bit disappointed when looking at those results because, well, labeling density was not as good as in the other setups. But keep in mind, and that's what I concluded a little bit later, that the primary antibody did penetrate into the cell cytoplasm. So tubulin fibers are being demonstrated over here, are being labeled. So the primary and also the gold reagent could penetrate, could enter the cell cytoplasm, could penetrate the cell membrane. So labeling of the cell interior is possible after fixation in aldehyde when you treat your specimens after the aldehyde fixation with sodium borohydride. So that's super important. And the fact that you can leave out detergents has a beneficial effect on morphology. So these specimens were incubated with primary antibody for 1 hour and incubated with gold reagent for 2 hours. What would happen if you extend the incubation time? Does this have a positive effect on labeling density? Here you see the same setup, so cells fixed in aldehyde, treated with sodium borohydride, but now I incubated the cells, the cover slip culture, with the ultra small gold reagents for 4 hours instead of 2 hours. I washed, silver enhanced, and then this was the result. So not only you could see an intense staining, a more intense staining in comparison to the incubation time of 2 hours with the ultra small gold reagent, but also the stretches of tubulin that are stained, that are labeled, are longer. So this means that more antigen will be available for detection in relation to the immuno-incubation time that you're using with your gold reagent. So to get sufficient penetration into your specimen, in general with pre-embedding, the incubation time with your primary, but certainly also with your ultra-small gold reagent, is long. Yeah, with cells, 4 hours is the minimum. With vibrotome sections, for instance, an overnight incubation is something that you need to get full penetration into the interior of your specimen. In many cases, after aldehyde fixation, the treatment of the specimen with sodium borohydrate is sufficient to get your reagents into the cell interior. Sometimes you need to combine this with also a treatment, a permeabilization treatment with Triton X100. And I recommend you to compare these setups and see which treatment works best. So in general, pre-embedding immuno gold silver staining, and that's what we recommend, is depending on the thickness of your material, so for either cover slips or vibrotome sections, 50 to 200 micrometer thick vibrotome sections, you start with a sodium borohydrate treatment, and you always use this method. 50 minutes for cover slip cultures, 30 minutes for vibrotome sections. You wash your specimen, and then depending on your material, on your specimen characteristics, also depending on the fixation that you're using, the fixation method that you're using, you may need to include a low concentration of Triton X100 to facilitate reagents to enter the cytoplasm, to enter organelles like mitochondria or the Golgi apparatus, and label antigens that are also present in those cell organelles. So always include sodium borohydrate and check, compare, see if Triton X100 is needed, yes or no. The use of detergents may have some negative effects on morphology. What we also have encountered is that when you use a phosphate buffer to dilute your chemicals in, so your sodium borohydrate and your Triton X100, when you use a phosphate buffer, the morphology is better than in comparison when you're using PBS as a substrate, as the buffer to dilute these chemicals in. So treat with sodium borohydrate and phosphate buffer, and check, test if you need the use of Triton X100 to visualize your and to detect your antigen in a pre-embedding setting, yes or no. And when possible, check results in the light microscope before embedding, so before the preparation for electron microscopy. You can see a lot of antigens already being visualized in bright field light microscopy, and if the concentration of your antigens is low, then check results in confocal. With a confocal microscope, you can visualize your single gold-silver nanoparticles. So this is the way to also check at the LM level if your detection has worked, yes or no. And if you have a positive result, the positive control, then afterwards embed these specimens or specimens that are simultaneously incubated this way, embed them in epoxy resin, section for EM, go towards the EM, and when you are behind your microscope, your EM microscope, then you are certain that there are particles there because you have already checked them in the LM beforehand. So incubation time with both the primary antibody is important, and incubation time with the gold reagent is important. In general, for pre-embedding, you need long incubation times. So at least 4 hours, and in general, overnight incubations are being used to get full penetration into a vibrotome sections of 200 nanometer thickness. micrometer thickness. And the reason, of course, that you need this long incubation time is that because of the fact that there are more proteins present in the cytoplasm, the way, the length the antibodies have to follow before they can reach the antigenic site is far more longer than in comparison to a specimen that is being permeabilized with Triton X100 during fixation, where you wash out most of the cytoplasmic constituents, most of the proteins. So if you need a long incubation time for the antibodies to get into the cell interior, to reach the antigenic site, then also washing steps need to be long. You can't expect to wash out in 3 times 5 minutes your unbound primary antibodies or gold reagent when you need incubation times of at least 4 hours to get full penetration into the cell interior. So if your incubation time needs to be long, then also washing times and the number of washing steps need to be long. That's something that you need to take into account in pre-embedding setup, in a pre-embedding setup. So to help diffusion, to help the effectiveness of washing steps, then agitation, shaking will be of help. And you can do that on a shaker, of course. If you have an automated system like the PrepMaster, then this is also a way to, this setup is also a way to promote washing, to promote incubation, to promote diffusion, and that will certainly be of help in order to help you with the in order to obtain a good result. What also is of importance is dilution of your primaries and your gold reagent. Use high dilutions of primary antibodies and gold reagents to sort of to have every antibody that is available, or most of the antibodies that are available for immunodetection, specifically bound to your antigen. And use high dilutions. In general, in comparison to a post-embedding setup, your primaries and also your gold reagent needs to be diluted 2 to 4 times more in comparison to those conventional setups, so these on-section labeling setup. That's something that may be helpful also in generating a good signal-to-noise ratio. A second, or not a second, but one of the last things that is of importance to get or to combine good signal intensity with a decent morphology is the use of osmium. Osmium that generates membrane contrast. Osmium also, or may also, have an effect on your silver particles. So osmium in time reduces or oxidizes your silver precipitate. So if you, and you normally want to use osmium to generate membrane contrast, if you use osmium, then use it at a low concentration, 0.5%, and either use it in deionized water or phosphate buffer for a short or relatively short period of time. 50 minutes for cells, max 30 minutes for vibrotome sections. If you use this post-fixation treatment for this amount of time with this concentration in deionized water or phosphate, then your particles remain there. So you will not lose signal using a setup with osmium for this amount of time and concentration. Do not use osmium in Caco-delay buffer in a pre-embedding staining. Do not use osmium in Tris buffer or sucrose, for instance. Setups that can be used just for heavy metal staining, but do not use it in combination with silver enhancement, because if you use these buffers, then in time you will lose your silver precipitase, you will lose your silver particles. You will lose your signal in EM. Osmium will not affect your gold label. So in those instances that you have the impression that your osmication had a negative effect on labeling density, then always silver enhance or do a silver enhancement, on-section silver enhancement, to see if those particles that are on the surface, gold particles that are on the surface of your specimen, can be silver enhanced again, and in this way you can visualize your signal. So that's something that you can do too. And using this setup, does this give you the option to label antigens within the specimen and combine this setup with a nice morphology? Yes, this is certainly optional. And we have also several examples presented on our website and presented in newsletters. The two examples that I want to show you now is the pre-embedding label of dopamine transporter protein on vibrotome sections of mouse brain tissue. And when you look at the specimen, you see the surface of the specimen over here, of the section, and you see labeling of the DAT terminal, so the dopamine transporter the terminal within the section, within the vibrotome section. You see label combined with a nice morphology. So mitochondria are very well preserved, the nucleus is very well preserved. A very nice way to combine labeling, immunolabeling, with morphology. And again, the setup is simple, straightforward. You have your specimen fixed in aldehyde, vibrotome section, and you label your vibrotome section with reagents before embedding in plastic and epoxy resin before sectioning. So a simple, straightforward method. Another example again of pre-embedding immunocytochemistry using again ultra-small gold reagents with silver enhancement. In this case, the labeling of a Golgi protein marker, NGP160. It's again work from Yi, Atlanta. And this antigen is located within the lumen of the Golgi. Yeah, so the antibodies, so primaries, the gold reagent needs to penetrate not only the cell cytoplasm, so past the cell membrane, but also needed to pass the membrane of the Golgi organelle. So morphology, pretty good. So the Golgi stack is clearly visible, membranes are clearly visible, vesicles are visible, ER is visible, all the constituents within the cytoplasm are visible, and the label is within the Golgi apparatus. Yi fixed in a combination of paraformaldehyde and glutaraldehyde. She treated her samples with sodium borohydride, and after that treatment with borohydride, she permeabilized in a low concentration of Triton X100 in phosphate buffer and incubated with primary antibody, secondary antibody, fab fragment in this case, coupled to ultra small gold, and silver enhanced. The particles were silver enhanced with Arion Argent SEM, embedded in epoxy resin and sectioned for EM evaluation. The complete protocol is presented again in this Arion newsletter, which can be downloaded from our website. So please feel free to do so if you want to have more info on this setup. Yes. Few years ago, we introduced 2 nanometer gold nanoparticle conjugates particles conjugated to goat anti-rabbit and goat anti-mouse secondary antibodies. And we feel this can be an intermediate between conventional gold and ultra small gold reagents. Due to the fact that the resolution of microscopes, EMs, has improved, due to the fact that there are options to, like EDX or high-angle annular dark field microscopy, these particles, it will be possible and expected will be possible to visualize these gold nanoparticles without silver enhancement and combined pre-embedding, high-resolution pre-embedding with Immuno Gold Silver Staining using 2 nanometer gold conjugates. Haven't seen these results yet, and we hope in the nearby futures, researchers will use these conjugates for Immuno Gold Staining, for pre-embedding and post-embedding Immuno Gold Silver Staining, leaving out silver enhancement. However, this slide is there to show you that also 2 nanometer gold nanoparticle conjugates are able to penetrate into the cell interior. And in this example, you see a vibrotome section of an organotypic brain culture, so slices that are between 100 and 400 micrometer thick that are being incubated with a anti-GFP antibody to detect GFP constructs present in this tissue. The anti-GFP antibody is being visualized using a goat anti-rabbit 2 nanometer gold conjugate and is being silver enhanced with the Arion argent reagent for electron microscopy. Again, after enhancement, the specimen is osmicated, embedded in epoxy resin, sectioned, and the thin sections are being evaluated in the EM. A last example of what you can do with ultra small gold reagents, and that is the detection of antigens in living cells. So not fixed in aldehyde, but really in a living cell culture. You can do this by using a ketonic lipid, so a charged lipid complex, DOGS, which can bind not only antibodies, but can also be incorporated into the cell interior. So it will pass the cell membrane, and then within the cytoplasm, the antibodies can be transported towards the area where the antigen is located, in this case, within the cell nucleus. So a Fab fragment directed against polymerase II was being, an antibody fragment, was being conjugated with ultra-small gold nanoparticles. And this complex of DOGS conjugated, or the complex with the ultra-small gold conjugate was incorporated within the cell cytoplasm, delivered to the cell nucleus, so the antibody could pass the nuclear envelope, the nuclear membrane, and detects the polymerase II present within the cell nucleus. After detection, and these are really long incubation times of 12 to 24 hours, the cells are being fixed in aldehyde, osmicated or silver enhanced, osmicated, embedded in epoxy resin, sectioned, and then these sections are being evaluated in the electron microscope. So label present within the nucleus. So that's something that may be of interest too. So next to the labeling of antigens, pre-embedding labeling of antigens in aldehyde fixed specimens, you may also detect antigens in living cells in cell cultures using this DOGS complex. So pre-embedding, it's again a straightforward, simple detection method that resembles immunofluorescent incubation methods or immunoperoxidase, enzymatic immunodetection methods. It's simple, straightforward. You don't need low-wick reels. You don't need any special plastics. You don't need any special equipment. It's simple, straightforward. You can use epoxy embedding. You have a hydrated specimen, and that's really an advantage. And not only the labeling, the surface labeling, but really hydrated specimen, you have the option to label all the antigens that are available. A disadvantage, or something that you need to take into account, is the incubation time. In general, you need long incubation times, overnight incubations, and if you need a long time to get your antibodies within the cell interior to bind to your antigens, then also washing time, the washing steps, the amount of washing steps, washing time is long. Take that into account and use high dilutions of antibodies to get good signal to noise ratios. With the use of sodium borohydride, you can obtain a very decent, very good morphology. That's really a step that you should include into your pre-embedding protocol. Depending on your specimen characteristics, you may need to use a detergent step and use Triton X100 at a low concentration in phosphate buffer. And this combination may also give you a nice, a good morphology in combination with a decent labeling density. So that's certainly an option that that I should recommend you to test. There are, of course, other options, other permeabilization protocols that may give you good results and that can be compared with the setups that we recommend, like, for instance, the use of saponin, the use of freezing protocols, so sucrose infiltration in combination with freezing and thawing again. These are options that you can use, but in general, what we at least recommend, or what we feel gives a good end result, is this combination of sodium borohydride with or without Triton X100. So I would like to finish this webinar with the option, or to give you the option at least, to test our reagents also in one of our master classes. In these master classes, you can bring your own specimens, your own primary antibodies, and test our reagents under our guidance, and see if the reagents really do what we feel they can do. Is that's giving you high labeling densities, give you good signal to noise ratios. And in these master classes, we really go into detail within the procedure. So explain every steps that is within the procedure, give you hints and clues how to optimize a procedure and when needed. And may be of interest, and we'll certainly invite you to contact us if you want more details about our master classes. And hope to be able to see you there in the nearby future. So if there are questions, James, then I think we now have the option to ask, and then I hope to be able to answer them. So.
Yeah, absolutely. Thank you so much, Peter, for your insight into the Immuno Gold Silver Staining. I know throughout your talk I had multiple questions sort of popping in my mind, but I would like to invite the audience at this time to submit questions. If you would like to speak and ask your question out loud, please raise your hand and we'll unmute you and allow you to do so. So to start things off, we have a question from an anonymous source, which is, have you used pre-embedding IGSS with bacterial cells, and do you have any protocol recommendations?
with bacterial cells and also with plant tissue, you have, of course, a barrier, which is the bacterial outer membrane. And if you want to incubate specimens like that, and also plant cells which have a cell wall, then you may need to remove those the cell wall or the bacterial cell wall, treat them with enzymes in order to be able to get penetration into the specimen itself. And these enzyme treatments will certainly have a negative effect on ultrastructure. So these are really the difficult specimen to work with. So animal tissue is more easy and more compatible with pre-embedding staining than bacterial cells or plant cells. That's something that you need to take into account. A sectioning technique may be of help. So you can open your specimen, then with the sectioning technique by using vibratome sections, that at least a part of the cells can be labeled, the interior of the cells can be labeled. But if you really have a cell wall present into your specimen, then you certainly need to use an enzyme treatment and remove part of your cell wall, and this may have a negative effect on the morphology of your cells. So a more difficult specimen in comparison to an animal cell.
Penetrating membranes, cell walls, it's always a challenge in any of the...
Well, again, the cell wall is difficulty. The membrane, that's something that can be done with sodium borohydride and the Triton treatments. Yeah.
Okay, moving on, we have Anita Christie, who I'll allow to ask their question out loud. You may go ahead. I believe you have the ability to unmute. If not, I see the submitted question we can answer here, which is, can...
Yes,
sorry.
Have you recommended any?
Was it?
Yeah.
Yeah, go ahead.
Have you recommended any protocols for human autopsy tissues? And also, can you send us today's presentation? Because I am on and off between work and presentations. So.
On that note, the presentation is recorded. I'll leave it to Peter's discretion if he'd like to share his slides.
Okay, appreciate James.
And the question, Peter, was, do you have any protocols for human autopsy tissues in particular on the Orion website, or have you worked with those tissues before?
On autopsy tissue, you can use a similar protocol. The disadvantage, of course, of this tissue is that the morphology from the beginning already on is, of course, suboptimal, and that's something that you just need to deal with. But the setup is similar, so you can use these specimens also in a similar way than vibratome sections or cells in culture. Certainly possible. Yeah.
Okay, thanks.
Basic protocol is the same.
Okay.
Thank you. So we have another question from Stanislav, which is how much difference in antibody penetration would it make to use nanobodies instead of the regular IgG ultra small gold antibody conjugates? So the influence of antibody size on this penetration.
Smaller molecules will penetrate more easily. That's something that's for sure. So nanobodies will more easily penetrate in comparison to a complete IgG or a fat fragment. So with respect to penetration, this is beneficial. With respect to affinity, a nanobody, of course, has only one binding area, and in general the affinity of those antibodies is or can be lower, at least in comparison to a primary with a high affinity. And this in practice may mean that you need to use higher concentrations. So nanobody in comparison to a well-performing primary with, or an antibody fragment with a Fab2 fragment with 2 binding fragments. And if this is true and you use these, or you need to use these higher concentration, then this may have a negative effect on background levels. So yes, penetration is better, but be aware of the fact that background levels can be higher due to the fact that the affinity is lower than in comparison to a well-performing Fab2 fragment.
Thanks, Peter. And this person also wanted to know whether you've used a microwave processor for incubation and washes before.
Yes, I have done that in the past also, but then in the setting, so in a post-embedding labeling setup, there I used the microwave. My experience, again, but it's my experience, was that the incubation times could be shortened, but that the background levels in general were higher. So I always got the best signal to noise ratio when combining a relatively long incubation time with a high dilution of the primary and the gold reagent at room temperature or at 4 degrees, in comparison to labeling or incubating in combination with the microwave. But again, if you put in more energy by using this microwave, then in general, incubation time will be shorter. That's true. But the nicest results, the cleanest results I get when I label at room temperature and use long incubation times in combination with a high dilution of the primary, the high dilution of the gold reagent.
We have time for a few more questions. So Michelle asked, and I think this was in response to your answer about bacteria, which was, with respect to C. elegans, do you need Triton to get to the cuticle? Have you worked with C. elegans before?
I haven't worked with C. elegans, so if I want to give a suitable answer to that question, it might be wise to just contact me via email, and then I'll check and can give a more optimized answer in comparison to what I can do now.
Thanks, Peter. Roberto has some 2 questions and has asked, how long would incubation need to be for thicker tissue sections, such as a half micron?
Half
500.
micron.
Sorry.
Again, it depends on, there is this combination of thickness of the tissue and fixation. So if you have a specimen that's only fixed in paraformaldehyde, treat it again with sodium borohydride to convert this sheath base towards a single bond which is stable. That's important. Then incubation times of, let's say, 6 hours to 8 hours in general are fine. But why don't you just do an overnight incubation? I think that always is sufficient with these kind of specimens. If you have aldehyde fixed material, then you may even need to increase the incubation time a little bit longer. But I would start with overnight incubations with both the primary and the secondary. So these are really long procedures. It's not the specimen preparation procedure that is long when using pre-embedding. It's the immunolabeling procedure itself. So due to the fact that you need an overnight incubation with the primary, overnight incubation with the secondary. And again, take into account that washing then also is something that is, washing time is something that is crucial. And to avoid the fact that you also need to use overnight washing steps, use high dilutions of primaries and secondary antibody gold reagents. So for instance, our ultra small gold reagents, we recommend dilutions of 1 to 50 to 1 to 100 of the reagent for post-embedding labeling and also for the pre-embedding labeling of cells in culture. If you want to incubate these thick sections, go towards dilutions of 1 to 200 to 1 to 400 and incubate. Be sure that everything that you give to your cells also reacts with your antigen of interest. That's something that can be helpful to reduce washing time, but incubation time is certainly needs to be overnight.
And the final question we have time for was, would a perfusion distribution method work for gold particles? So can you perfuse along, after your fix or something, and wash with buffer and perhaps add the gold then, or maybe even upstream of fixation, or I'm not sure.
So if you have, let's say, your tissue and then do a perfusion, similar to what you do with the fixation, with the perfusion fixation, this setup, that's what you mean. If it's fixed in aldehydes, then you need to do something in addition in order to get penetration into your tissue, which is this sodium borohydride treatment, which is maybe also Triton X100 for permeabilization. That's of course something that is difficult when using this setup. So I always would, after the fixation, perfusing fixation, is recommend you to prepare, for instance, vibrotome sections from the area of interest, because you need something in addition to the chemical fixation to get really your reagents within the cell interior, within the cell cytoplasm. It will be different, of course, if you want to detect something that is on the outside of your cells, but in general, you need something in addition next to your fixation. But okay. You can always give it a try, of course, and see what happens.
Thanks, Peter, for that. And I will just add on to that last point, as someone who's done a lot of Immuno Gold pre-embedding, that there's a set of protocols that have been developed for folks working specifically in the eye, but I know it's also been employed successfully in brain tissue, which is the use of tissue expansion protocols. Okay. So including, you can't necessarily alter the extracellular space using ionic buffering, so by increasing your salt concentrations. But if you add in sucrose or sugars, you can actually start to tamper with the space between cells. And some argue that, especially in the brain, when you've gone through fixation and sort of gone through a standard EM protocol, that end result that you see where all the neurons are densely packed and there's really no extracellular space is a kind of artificial product of the process. But if you upstream of fixation or just downstream of fixation, you treat with sucrose, and there's some methods available out there for this, you can actually preserve that spacing. And so things will look quite strange if you're not used to seeing it, but you'll see larger amounts of extracellular space. And this has been extremely beneficial in applications where antibodies are being used to help get them perfused through tissue sections. So perhaps in relation to the person who asked about the 500 micron section, you could consider these extracellular space expansion or preserving protocols. Okay, so with that, I'd like to thank Peter for his time today. And I apologize, I misspoke and said Orion was in the UK, but it's in the Netherlands, and has always been. So excuse that mistake here. And we would like to thank everyone for hanging in there. We apologize that it's run a little long, but we appreciate your time. And if you have any follow-up questions, we will reach out with the recording link, and perhaps Peter will share his contact information so you can reach out to Peter directly to get some additional advice. And you can always visit his website. And yeah, sure, go ahead.
Contact details are also in the presentation, but just visit our website. General questions can already be asked to our info@orion.nl address, and then they will reach me too. But you find my contact details within the presentation, but also on our website. So, and otherwise, just contact EMS, and they know how to reach me too. So,
perfect.
That's an option.
Yeah? You can always ask us. We'll get you to Peter. We'll get you the answer. So, Peter, thank you so much, everyone. Thank you so much. Have a great rest of your week. Thank you.
Thank you.